Hi everyone! Today we will begin the three-part series on the cardiovascular system, which is divided into three chapters. One is about the blood, one is about the heart, and one is about the blood vessel. For chapter 19, we will begin by talking about blood, which is again a pretty straightforward chapter about all the cellular and protein components of the blood. We'll go through some how blood typing works and why certain transfusions can't happen between people with different blood type, and how can we figure out someone's blood type. Now, the most difficult chapters are the next two on the cardiovascular system about the heart and the blood vessels, because they involve a lot of mechanisms and even some physics, relationships, and values. But yet these are all very interesting chapters. So, let's enjoy learning about them. Okay, so let's get started. As we learned from the tissues chapter last semester, we know that blood is one of the specific kind of fluid connective tissue that contains red blood cells, white blood cells, and platelets, and all of them are suspended in a fluid matrix called the plasma. So now we will go into details about each of these individual components of the blood. We kind of glossed over it in the tissues chapter last semester. Now, as usual, each organ system chapters begin by going through some of the main functions of the system. So our blood is in charge of transporting dissolved gases such as oxygen and carbon dioxide. And this is kind of like a major theme with cardiovascular system and respiratory system that we'll talk about. So be ready to hear a lot about oxygen levels and carbon dioxide levels, what our body does to when we don't have enough oxygen, what happens when we have too much carbon dioxide, what happens. The blood also does regulation of pH levels and ion composition, for example, calcium, potassium within the interstitial fluid. We'll get to the pH stuff. How blood is able to serve as a buffer for those different pH levels of the blood when we get to the chapter on respiratory system. That's when we bring in the pH regulation part of it, because it has to do with how much carbon dioxide there is in the body. Okay, so that's why they save that for the respiratory system. Okay, they also restrict fluid losses at injury sites by initiating the process of clotting. So this is a detailed process that involves a lot of stuff and a lot of steps. If you ever need to take an immunology class, there will be a lot more steps that you will have to know for clotting. But here, the last part of the chapter will kind of summarize the process of clotting into three phases and just be able to name a few of the clotting factors that's involved. Okay, and then, of course, the part of the blood, there are white blood cells, and white blood cells' main job is to defend our body against toxins and pathogens. Whatever foreign invader that doesn't belong to our body, they will fight it. Okay, some use the cells, the cell themselves can fight the pathogen. Some will need to make antibodies. So we'll talk a little bit about that here, but we're going to get to the bulk of that when we get to the immune system in a later chapter. And we already learned from the muscles chapter last semester that, the blood also stabilizes our body temperature by absorbing heat from our muscles. And that's why when our muscles shiver in contraction, it generates heat. That heat gets transferred into the blood surrounding the muscle, and hence that's how we are able to keep ourselves warm in some fashion. And that's why, vasodilation and vasoconstriction is something we'll talk about when we get to the blood vessel chapter. So, and the blood has these specific following characteristics, and you have to know these. Make sure you know these characteristics of the blood. The temperature of the blood is actually slightly warmer than body temperature, not by much, just by a degree Celsius. So they do average about 38 degrees Celsius, which is why you when you feel blood is pretty warm coming out of the body. And blood is about 5 times as viscous as water because there's all those components in here that you can see. There's cells, there's many different types of cells, and also there's a lot of proteins that's circulating and suspending in this yellow looking liquid that you're seeing here called the plasma. So that's why it's not the same as water. And on top of that, there's ions, salts, electrolytes, etc. Lots of stuff is in our blood, and that's why blood is the main transport system for our body. Blood is actually slightly alkaline, and this is a pH value I do want you to know because eventually you will have to know it anyway. We keep bringing this very narrow range of pH level that your body has to keep at. We'll talk about it in respiratory system. We'll talk about it in the urinary system. We'll talk about it in the fluid electrolyte chapter. So you'll see this through, multiple times throughout the semester. But we have to keep our bodies in this stringent, narrow range of pH. If it's too high or too low, we can have the case of acidosis or alkalosis. And we'll get to those when we talk about the respiratory system and also the fluid electrolyte chapter. Okay, some stems from respiratory means, some stems from metabolic means. Okay, so the blood volume and the total blood volume that's estimated in an adult individual is usually, we can have a good rough estimate by this formula right here. Okay, the total amount of liters of blood that a person should have, it should be about 7% of their body weight. Okay, so if a person weighs 50 kilograms, then you just need to multiply that by 0.07 to get how many liters of blood that person is supposed to have in terms of their total blood volume. So this is just a rough estimate to remember. All right. So the two main components of blood, like I was saying, one is the liquid portion and one is the cellular portion. The liquid portion is called the plasma, okay? And it makes up about 55% of your total blood volume. What's in the liquid are plasma proteins mainly, and they are mostly synthesized by your liver. And which is kind of what I was talking about last chapter, how the liver has a foot into everything, including all these proteins that's circulating around the blood. Now, there are 3 major types of proteins that's floating around in your plasma liquid portion of the blood. Albumin is what makes up the majority of plasma, which is why they are the major contributors to plasma osmolarity and osmotic pressure, because there's so many of them. And again, the majority of these proteins are made within the liver. So albumin can include transport fatty acids, thyroid and steroid hormones that we talked about last chapter, etc. Now, the second most abundant protein group within the plasma is globulin. And these include immunoglobulin, which are antibodies that is produced by our immune system, of course, which is why there's such thing as plasma donation versus whole blood donation, because those proteins within the plasma can be very helpful to another person that has some rare genetic disorder, for example, that require certain clotting factors, antibodies within the plasma that can help that particular individual. Now, the other type of globulin are transport globulins, which includes these 4 different types of transport globulins. Some are hormone binding. For example, we talked about thyroid hormone binding globulins last chapter with the process of making thyroid hormone. And once they're done, they have to bind to transport proteins. And one of those is hormone binding protein. Metalloproteins we'll mention later on in this chapter, but they usually bind to metal ions. Okay, so for example, iron is a major metal element that's in blood. And the specific metalloprotein that carries iron is called transferrin. Again, I'll come back to this idea later in the lecture. Apolipoproteins are for moving triglycerides and lipids. And just the name itself, apolipo, kind of reminds me of adipose tissue. So to me, that's how I relate apolipoproteins to some kind of fat and lipid level transport. And the last type is steroid binding proteins, such as those that, bind to testosterone or, estrogen. Those are steroids, and they have these binding proteins to keep them around longer within your circulatory system. All right. And the last type of protein that's within the plasma that is critical for the normal blood clotting process is when we're injured, for example, are called fibrinogen, okay? Which is a precursor that gets converted into fibrin to close up your wound. We'll talk about the detailed process of clotting as the last concept of this chapter. So we'll come back to this at the end, okay? The other main component of blood, which makes up the other 45% of whole blood. Now, make sure you remember these percentages right here. Percentage of plasma and the percentage of form elements. That should be, the range a normal person's blood should fall under, okay? Your lab manual and lab exercise actually goes into more detail and separate the range between males and females. So for lab practical purposes, make sure you know the range percentage of plasma versus form elements for the lab between male and female, okay? All right, so this 45% of form elements is the cellular components, okay? And the cellular components, of course, includes your red blood cells, white blood cells, and your platelets, okay? Now, there are lots of, different numbers throughout the semester, and usually I don't ask for statistical things. So, but there will be cases where I will specifically say to remember these numbers because it's a number that's important and relevant to the material being studied. And for this form element percentage number, you will need to know it for the, like I said, the lab practical and the lecture as well. So, make sure you remember this. But what this portion of the blood contains are those that make up the cellular portion of the blood, okay? And we'll talk about each of these cells in detail. And in addition, all of them are produced in a process called hemopoiesis, okay? So this is another good word to remember, okay? From last semester, I already talked about how if a word begins with hemo, it usually has something to do with blood. Now, the other word I want you to remember is poiesis. Poiesis tends to deal with the process of generation of something. Okay, so there's always something poiesis, another something poiesis. Okay, so poiesis is the generation of, and whatever it begins with, then it's referring to generation of that particular part. So for this case, hemopoiesis has to do with generation of blood. And we're going to separate into erythropoiesis, leukopoiesis, and thrombopoiesis for generation of different components of blood for the form elements, within the form elements. Now, hematocrit is a diagnostic tool that is used, and you'll actually learn about this in lab as well. It's a normal clinical measurement of form elements that is your cellular component volume compared to the whole blood volume that can provide some clues for maybe a pre-diagnosis of the patient's situation at the moment. And I'll talk about, what these situations are on the next slide. But hematocrit is not the number for your plasma level, okay? The hematocrit percentage do have to do with the percentage of your formed elements, okay? And hence, wherever your blood concentrated cellular volume is, that is your hematocrit percentage, okay? And I'll go over that in this slide. All right, so 99% of the form elements portion of whole blood should be made up of red blood cells, or the correct medical term, or what I like to call the fancy term, is erythrocytes. And these are tiny little cells because one drop of blood alone, there can be 260 million red blood cells. Now, this is the type of statistical number that I don't expect you to know or have to remember because it's not really relevant to the material or the concept we're learning. It's just a number to give you an idea of how many red blood cells there are in our blood. So, don't waste your memory space on these type of numbers, okay? And so the color of the blood being red is actually due to the presence of a specific protein called hemoglobin. And it is because of the existence of this protein in blood that allows for binding and transportation of respiratory gases such as oxygen and carbon dioxide, okay? So this is a very critical protein that helps blood do what it needs to do. Now, and hematocrit can be used to see whether a person has enough blood, enough red blood cell particularly in their body, because 99.9% of that form element is your red blood cells. Now, you would never be able to see a big portion of white blood cell in the hematocrit unless something's wrong with the person's body, okay? So usually you see a whole red portion of the hematocrit like this after you spin down the patient's blood, okay? So how this diagnostic test is done is first you will obtain a blood sample from the patient, and then that sample will then be centrifuged with a high-speed centrifuge, of course, and it separates the cellular portion at the bottom half of the tube right here. And of course, why they settle down to the bottom of the tube is because they are more dense. Whenever you spin things down, they kind of settle according to their density value. Okay, and cellular portion is more dense than liquid portion. So the top half then will be your plasma liquid portion, which is that 55% we were talking about. So volume of red that we're seeing compared to the total volume of the top half here. So this is the red, this is the top half with the plasma. This portion, that percentage of red compared to the entire volume, is what we refer to as the percent hematocrit, which tells us how much of the patient's blood is actually made up of erythrocytes, which should be close to that 45% I was talking about earlier on average in a normal, healthy individual. So what does it mean when we have abnormal hematocrit reading? What can it potentially indicate? Now, if the hematocrit reading is higher than what it should be, and that's what here this increase mean. If it's more than 45%, then it can indicate two things. It could mean one, they're dehydrated. This person does not have enough water, which means their fluid level will go down. So this plasma level here would go downwards. So that when this plasma fluid level is not as high as it should be, even though the cellular component didn't change, but it ends up taking a bigger portion of the total volume because the person no longer has enough fluid portion in their blood. And that happens when a person is dehydrating. Okay? Or the second situation that can lead to a higher hematocrit reading is if there was something called erythropoietin stimulation. We'll talk more about this hormone in a bit, but it is a hormone produced by the kidneys when it thinks there's not enough blood. Now, if there was a greater, or if somehow you need more oxygen, okay, your body has more oxygen demand, that will require more blood as well, which then will also stimulate your kidney or your liver to release this particular hormone for production of more blood. Okay, in that case, we're not changing the liquid volume. We're not changing the liquid volume. What we end up changing is the form element's volume and raise it up. okay? So that now we have a higher percentage of hematocrit than what it normally would be. So either in this case, whether you're increasing cellular component or whether you're losing the fluid amount, will also, both of the case will indicate a higher hematocrit percentage, okay? So that's why it can be either thing. We'll talk about the application of EPO later in lecture. On the other hand now, if there is a decrease in hematocrit level, which means it is lower or less than 45%, that could mean, again, two things. Either there's internal bleeding, so the person is losing a lot of erythrocytes from that, or there could be something going wrong with erythrocyte production process called, like I said, the hematopoiesis, where not enough erythrocytes are being produced, okay? It could be because EPO doesn't function as they should, or the kidney or the liver is not producing EPO, or something is wrong with the process of regeneration of red blood cell that can lead to a lower hematocrit. Regardless, this person just doesn't have enough red blood cells. That's why you end up with lower amount of hematocrit, okay? Or if they have been bleeding severely. Now, structurally speaking, erythrocytes takes the shape of a biconcave disc with a central region that is thicker on the outer edges, okay, compared to the inside. It has this nice curvature if you look at it from the side view. And of course, that's actually useful for a couple of reasons that I'll talk about. But the structures of having these curvatures and capability of being flexible is what allows erythrocytes to have these specific 3 different types of function for blood. One, whenever there's extra curvature, just like how I talk about with microvilli, those curvatures are there to give a larger surface area to volume ratio, okay? Any curvatures is always more surface area than a straight line. So if you, for example, if red blood cells are straight boxes like this versus this, okay, you will have a lot less surface area in this case compared with surface area in this case. So that's why red blood cells have this particular shape for their purpose of faster exchange of gases that's allowed, okay? Now, in addition, that biconcave part of the red blood cells, what enables red blood cells to form stacks, okay? So if you look at some microscopic images, or in this image right here, you may be able to find these stacks of red blood cells like this, where they're all lined up closely next to each other. And we usually call those ROLU, okay? So ROLU stands for stacks of red blood cell. And it is what allows them to have that smooth blood flow through very, very narrow blood vessels. When we get to the blood vessel chapter, I'll talk about diameters of arteries, veins versus capillaries. And some capillaries in our body can be as thin as your hair, okay? One strand of your hair. And that's how sometimes because how thin it is, only one red blood cell diameter width can go through. So that's why the stacks of red blood cell like this then becomes very helpful for moving those red blood cells along through, okay? Because we cannot have anywhere in your body that blood cannot reach to or get to, or else we can't deliver nutrients and oxygen to that area. So ROLU is what this particular shape of red blood cell enables them to form. Now, the last thing is this particular shape having these curvatures and not solid block of hard plasma membrane is what allows red blood cells to bend and flex when they're entering those small capillaries. So imagine if you have a circle of blood vessel. Okay, this is the diameter of the blood vessel. Red blood cell can actually squeeze through like that, okay, because they have that curvature and they have that soft plasma membrane that enables them to kind of curve that way into very, very tiny, small spaces of the capillaries. Now, the other thing that is different in erythrocytes compared to other typical cells is that they are anucleate, which means they don't have any nucleus. Okay, almost every cell has a nucleus, and their precursor actually have a nucleus. Before a red blood cell become a mature red blood cell, they did once had a nucleus, but once they become a mature red blood cell, because we want them to be focus on what their function is for, which is transport of important gases, we remove the nucleus from the red blood cells. In the end, they only retain the cytoskeleton of the cell during the maturation process, and I'll talk about that whole process of erythropoiesis in a bit. So that's why I have this funny comic here in pretend conversation that would happen between a white blood cell and red blood cell, them making fun of each other because of what their function is. Okay, all right. So erythrocytes also have a very short lifespan compared to other cells. Okay, they usually cannot live longer than 120 days. So their lifespan usually is equal to or less than that. Hence, this is why they need to be constantly regenerated so they can be replenished and replace those that die off. And we'll talk about the process of erythropoiesis, which is erythrocyte generation and maturation, in a bit. Now, we mentioned that what gives erythrocyte their color and capacity to transport gases is the protein hemoglobin. Well, it turns out that the protein structure of hemoglobin is what gives its unique structural pockets that will bind to those oxygen molecules or carbon dioxide molecules, because they need to be able to bind both, okay? Because at different areas of the body, you are dropping off oxygen, and different parts of the body, you are dropping off carbon dioxide. So one hemoglobin molecule is actually made up of 4 polypeptide chain, which I think you probably have learned before in your other general biology classes already. Is the classic protein that almost every biology book uses to introduce the macromolecule protein. When we talk about the different level of structure, primary, secondary, tertiary, quaternary. Hemoglobin is the classic protein they use to explain quaternary structure. One hemoglobin is made up of 4 polypeptide chain. 2 here in the dark blue is called alpha chain, and 2 here is called the beta chain. And a single molecule is embedded within each of these chain here, these red penny looking thing. That is what we call a heme group. Okay, and heme group is made up of all these different chemical structure, and it has a molecule of iron ion in the middle. Okay, so it has this ring structure that specifically binds to iron, and it is this heme group that allows hemoglobin to bind to oxygen. Okay. Now, it is important to note that the interaction between oxygen and the heme group of hemoglobin needs to be weak. Okay, it has to be a weak relationship because, let me highlight that, weak relationship, because we don't want oxygen to be permanently bound to hemoglobin. Okay, that's not the purpose of blood, is not just to bind oxygen and keep it for life. Okay, it needs to have a way of acting as a transportation purposes. Okay, it needs to bind oxygen easily, but it also needs to release oxygen just as easily as well, or else your hemoglobin red blood cells are then going to pass by your tissues, and then the red blood cell is like, no, I want to keep this oxygen to myself, I don't want to dump it to your tissues. Then your tissue starts lacking in oxygen, become deoxygenated, and then they start dying. So that cannot be the case, okay? Which is kind of a side note here, is what carbon monoxide does when people smoke, okay? Why smoking individuals ends up not being able to do really intense sports, and they, kind of easily lose their breath, is because carbon monoxide binds at this very same spot of heme groups, just as oxygen and carbon dioxide do. But the binding relationship is a lot stronger than what it is with oxygen and carbon dioxide, because it's not meant for us to have carbon monoxide in the body. You ingested that poison through smoking, cigarette smoke. And hence, what it ends up doing is that it takes up all the spot that you have in your hemoglobin that's supposed to bind to oxygen. It just binds there, if I remember correctly, I think it's 200 times as strongly as oxygen. So you end up keeping that spot and taking up that spot. So now you can't use that particular hemoglobin for oxygen molecule delivery. And that's why for individuals that smoke, chronically, they would end up, easily running out of breath, and they can't really participate in really intense sports, is because their body loses the capacity to deliver those oxygens and bind to those oxygen molecules. All right, so when oxygen is bound to hemoglobin, it is called oxyhemoglobin, okay, which gives that bright red in color when we see oxygenated blood. Now, when oxygen is not bound to hemoglobin, it's called deoxyhemoglobin, which is then changes the color to dark red. It's not blue, okay, but people tend to refer to it as blue because we see veins as blue, but it's actually not blue in color. We don't have blue blood on like aliens do, if aliens exist at all. But we have dark red blood, which is when hemoglobin is not bound to oxygen. The way that the light is reflected off of that protein structure without a bound oxygen is what changes the color that it appears to our eyes. If you remember from your general biology class, what we see as visual colors of objects is based on what wavelength of light is reflected back to our eyes. And hence, that little structural change that hemoglobin goes through makes that reflection a little bit different. And that's why then we see a little bit darker red color when there is no oxygen bound to hemoglobin. Okay, so that's why depending on oxygen content of the blood, it can appear in different shades of red, which explains, for females, explains your differences in blood color throughout your menstrual cycle. How in day one, you start with a little bit brown, and then it starts to get dark red and really red, and then, goes back to the cycle of, go back to brown color when you're almost done with your period. Okay, that's all due to the amount of oxygen that's in the blood itself. Okay. All right. So interestingly, when we are all still only a baby in our mom's womb, we have a special version of hemoglobin called the fetal hemoglobin. And what's special about this type of hemoglobin is that it actually binds to oxygen molecules more tightly than adult hemoglobin. Not as tightly as carbon monoxide that I was talking about. It just has a higher affinity for oxygen than your normal adult version hemoglobin. Now, why this is important is because it is what enables and ensures that oxygen is delivered to the fetus in adequate amounts. Imagine if both fetus and mom's blood have the same affinity for oxygen, then how can we control and make sure oxygen gases gets dropped off to the baby's blood through the placenta? What if that blood just goes right by and not drop off enough oxygen? Okay, so there's no special pump that can ensure that oxygen is going to be pumped towards the baby. Okay, so in order to be sure, because when a mom's body is forming an embryo and a fetus, everything goes to prioritizing of the development of the baby. Okay, and that's why I talked about last semester how pregnant moms need to ensure that you have enough calcium in your body, because when you don't have enough calcium, well, your body's main priority at the moment is form those baby's bones. So it will take your calcium away from your bones and to form the baby's bones. Same thing here. The main point of the mom's body when it carries an embryo or a fetus is to supply everything that you have, okay, for enough for the babies to survive, okay, in the mom's womb. So that is why the baby have this special form of hemoglobin that can acquire those oxygen molecules to be sure that we're not just, we'll give 50% to the fetus, the oxygen, and then leave the rest back to the mom, okay? Then the baby doesn't have enough oxygen. You need oxygen for the baby to grow. There's a lot of cellular respiration and ATP that's required in the baby. So that's why they have this special hemoglobin to ensure all of the baby's hemoglobin also is saturated with oxygen. Okay? All right. But all this special fetal version of the hemoglobin will then actually be converted to adult form of hemoglobin from the time the baby is born to the time they're about 1 year old. Okay, so you do end up losing that hemoglobin because then you don't need it anymore when you grow older. You can breathe on your own and you don't have to rely on someone else to supply you with oxygen as a normal individual. And hence, knowing this concept has led to a clinical treatment. That's why I said this is interesting, because how we come up with clinical treatment and how we come up with new ways to treat diseases that we couldn't is from study of the human body. If we don't know how the body works, how can we come up with ways to treat things, okay? And because of these things we learn about the human body, then we come up with, so how can we mimic what the natural body does to help patients that has some certain inability to do something. And in the case of patients with anemia, they don't have enough red blood cells, okay? And there's actually a genetic disease called sickle cell anemia, which means that they make a different version of hemoglobin that makes the red blood cells look a little bit more sickle in shape instead of that nice round biconcave shape I was extensively talking about earlier for that nice structure of erythrocyte for those important functions. So this makes oxygen binding not very efficient for erythrocyte. Okay, for sickle cell anemia patient, their red blood cell actually seriously looks like this. Okay, they look like a little sickle, or they look like this. Elongated, okay, flat like that instead of nice and round circle that we see with a lot of red blood cell. So you can see how something like this, you lose that surface area we were talking about, okay? And that's why they don't end up with enough oxygen. And the other thing these weird shaped blood cells does, which is a big risk for people with sickle cell anemia, is blood clotting. Because remember, we talked about the shape of the nice round red blood cell is for them to able to squeeze through nicely through those tiny little capillaries. But when it's this odd shape like this, they end up stacking on top of each other in clumps, okay, instead of being able to pass nicely through the capillaries. So people with sickle cell anemia, not only are they lacking in oxygen, but they also are very much prone to blood clots. So they have to be on blood thinner of some sort too, to stop formation of blood clots. Okay, so a lot of problems goes with this genetic disease. Okay, so in order for the normal portion of erythrocytes to have more affinity for oxygen, because thankfully, usually those individuals that can survive through sickle cell anemia means, yes, they have sickle cells, but they also have a good portion of red blood cell in their body that is this normal round shape, okay? So the question is, how can we increase the affinity for oxygen in these round, normal shaped red blood cells? Well, they found out that, these patients, if they're treated with drugs, either hydroxyurea or butyrate, okay? If they're treated with either of these medications, it can actually induce an adult to make fetal version of hemoglobin in their blood. How cool is that? So that way they can at least have a higher attraction or affinity for oxygen molecules, even though they don't have enough blood. But maybe, all four of these pockets will be saturated with oxygen most of the time, because now you have this fetal version of the hemoglobin. As usually, hemoglobin floating around in our red blood cells are not entirely 100% saturated. So by increasing the ability of these red blood cells to be saturated with oxygen, we can actually deliver more oxygen to other, peripheral tissues of the body. Okay, so that's interesting how something we learn about in babies, we can use that same concept to treat individuals with certain diseases. Okay. All right, so each red blood cell contains 280 million, hemoglobin molecule. That's a lot, We're talking about one drop of blood already has a whole bunch of red blood cells. And in one of those tiny little red blood cells, there's 280 million of these hemoglobin molecules. And because one hemoglobin has 4 heme units from the 4 polypeptide chain, technically speaking, one hemoglobin is capable of binding to more than 1 billion molecules of oxygen, And yet you can see why we're still doing all these things to ensure enough oxygen delivery. You can see how much of a need we have for oxygen. We technically have all these billion molecules of oxygen capacity in all our blood, but yet we still run into all these problems, with when people have diseases and stuff. So that tells you how much our cells in every single part of our body is screaming for oxygen, and they need all those amounts of oxygen. Now, and the amount of oxygen bound to hemoglobin depends how much oxygen is available within the plasma itself, because what happens is the oxygen will actually diffuse from the lungs into the fluid portion of the plasma, and then they can bind to the hemoglobin proteins within the red blood cells when it pass by through your lungs. We'll review this concept in detail again when we get to the respiratory system, but for now we'll just do a quick gloss over it. So in peripheral capillaries, peripheral meaning blood vessels that are in other tissues and organs other than your heart and lungs, we deliver oxygen to our peripheral tissues. So the oxygen content in blood coming from peripheral capillaries are usually very low. So low oxygen concentration in peripheral capillaries. But after oxygen is dropped off, the blood will pick up blood coming from peripheral capillary, after oxygen is dropped off, the blood will pick up carbon dioxide as waste. So you see here, this is representing that of peripheral capillaries. So oxygen diffuses into your tissues, and as a byproduct of cellular respiration, if you remember, you have carbon dioxide generated in cells. So those will then be dropped off back onto the blood, so blood can carry the carbon dioxide back to the lungs. All right. So carbon dioxide levels will be high in blood coming from peripheral capillaries. So hemoglobin that is bound to carbon dioxide is referred to as carbaminohemoglobin. So you have oxyhemoglobin and carbaminohemoglobin. You tend to have more carbaminohemoglobin coming out of peripheral capillaries. On the other hand, in lung capillaries, blood vessels that surround those individual alveoli within the lungs. So this is the lung, for example, and there's millions of these little grape-like cluster of tissues that we'll talk about in the respiratory system, and also something we mentioned in the tissues chapter back from AMP1 when if you remember simple squamous epithelium, one of the examples we looked at was alveoli in the lungs. So this is what we're looking at when we're looking at those tissues back then, and hence usually these alveoli have a lot of these capillaries surrounding that whole cluster of spheres, is because the air goes into the alveoli when we breathe in, and then gases can travel across from the alveoli to those capillaries, and within those capillaries, of course, are your red blood cells that will pick up oxygen and drop off carbon dioxide. So we can pick up more oxygen, deliver it to your peripheral tissues, and also drop off carbon dioxide, so we can breathe out those carbon dioxide via our lungs and airway. All right, so Remember, lung capillary oxygen level is high and carbon dioxide level should be low because you just dumped it off into the alveoli. So hence anemia, as I have mentioned earlier, is a condition when there is not enough red blood cells within the body that can lead to a low hematocrit reading, or if hemoglobin protein itself was not abundant in red blood cells, like in the case of sickle cell anemia, then this usually leads to insufficient oxygen supply. Okay, and what I mean, why sickle cell anemia ends up with not having enough hemoglobin is because their hemoglobin protein structure is what messes up the whole thing. Okay, usually people with sickle cell anemia have, one type anyway, has just one nucleotide difference in their gene that encodes for the structure of hemoglobin. And just because of that one nucleotide difference, it results in the failure of that protein to fold correctly, and hence that's why they don't have normal hemoglobin, and hence that's why they can't bind enough oxygen and they don't have enough oxygen supply. Okay, but the resulting symptoms of anybody with anemia usually is they will feel weak, lethargic, and sometimes even confused due to the fact that they're not delivering enough oxygen to the brain itself. Remember, the brain is always the top priority of all the organ system. When we talked about neurons last semester, we said, glucose for sure have to be delivered to the brain. Oxygen for sure have to be delivered to the brain. Once the brain shuts down, everything else shuts down. So that's why the brain is always, or the nervous system as a whole, is always priority in terms of oxygen and nutrient supply. So we talk about red blood cells having a limited lifespan, which means they have to be continuously regenerated from stem cells. And this process is referred to as erythropoiesis, specifically for red blood cells, because urethro right here represents red blood cells, because erythrocytes. Okay, remember last semester I talked about osteocyte. Cyte usually means some sort of a cell. Okay, so urethro tends to mean something that has turned red. Okay, kind of like eurythmia. We talked about erythema when you get first degree burn, and erythema means areas of the skin that turns red. So that's why it's called erythrocyte for red blood cell, erythropoiesis for generation of red blood cell. Okay, and this process mainly takes place within the liver and the spleen during embryo development, but in adults, regeneration of red blood cells mainly happens in the red bone marrow. Now, there are specific type of stem cells for blood, just like how we had specific stem cells for bones. Remember those osteogenic bone cells we talked about. So for blood, their specific stem cells are called hematopoietic stem cells. Again, the name itself speaks for what it does. Hemato, okay, means blood. And then poietic, I already said something with poiesis, poietic, they have to do with generation of. So the name itself is generation of blood, and these are stem cells. Okay, so that's why the name itself speaks for what it does. And they can be, of course, abbreviated as HSC as well. But like I said, with endocrine system hormones, when you know the full name of something, it really gives you a good idea of what it does and what it's supposed to be for. Okay, so you can remember it as HSC if you're very good with letters and sort of relating to what those letters mean. Okay. So we have discussed before that in addition to red blood cells, there's also different types of white blood cells. So hematopoietic stem cells divides into two main groups of stem cell. One is called myeloid stem cell, which produces all the red blood cells in the body and platelets, and also some types of white blood cell. The other group of stem cell is called lymphoid stem cell, which are mainly for regeneration of a specific type of white blood cell called lymphocytes specifically. Okay. And I'll show you a chart later on in the lecture that goes over the breakdown of what hematopoietic stem cell produces. Kind of breaks it down into a chart format for what is the different stem cell branches that become your red blood cell, your different types of white blood cell, and also your platelets. Okay. So for myeloid stem cells, they will further differentiate into different what's called erythroblast stages, as illustrated in this particular figure here. There's pro-erythroblast, basophilic erythroblast, polychromophilic erythroblast, normalblast. I don't expect you to know all of these little steps. There's with A&P, there's just so much information, and I try to just narrow it down to what's the big picture, the gist. Okay, so for me, as long as you know from myeloid stem cell, they will differentiate into different stages of erythroblast. That's good enough for me. You don't have to know every single one of these little steps. Okay. And what you need to know is, after the erythroblast stage, you have to become another type of cell before you become a mature red blood cell. Remember, we mentioned earlier that red blood cells are anucleate, which means somewhere along the way of this process, they have to get rid of their nucleus. So that's exactly what happens here, where you have the erythroblast get to this day 4 period, and then boop, out comes ejection of the nucleus, and then it will become what's called reticulocyte. Okay, so this is the reticulocyte stage, where it no longer has that nucleus we were talking about, and they will go into circulation and spend another day there before becoming a mature red blood cell. Okay, so the flow goes HSC, hematopoietic stem cell, to myeloid stem cell, to erythroblast, to reticulocyte, and then your mature red blood cell. Okay, so I would really suggest for you to make a chart or something that does that. Okay, so ultimately at the top is you have HSC, hematopoietic stem cell, that becomes myeloid stem cell or lymphoid stem cell, and we'll talk about what that become later in lecture. And myeloid stem cell actually makes different stuff, a whole bunch of stuff, but one of it is that it will become erythroblast stages, which will then become reticulocyte and eventually become a mature red blood cell. Okay, and we'll talk about what these are later in lecture when we get to the formation of white blood cells. So this is a good, I always tell my students, a good way to attack all these biology classes is draw diagrams. Okay, draw these charts and then see if you can actually draw these charts from memory. If you can do this, then you can do the same thing on exams. Okay, it's hard to memorize all these words from a textbook or from even from my slides. Okay, but when it's narrowed down to something like this on a diagram, it makes it seems a lot easier to attack and memorize. Okay, all right, so that's the flow right here for erythropoiesis. Okay, all right, and we'll talk about the other poieses later in the lecture. Let me go back to this. Okay, I keep forgetting to press the chart button instead. Okay, all right, so the whole process of generating red blood cell is stimulated by a hormone, of course. That's why we begin the whole semester with endocrine system, because almost all processes in the body have to be regulated by some sort of a message. Just like how we need to turn on the light switch in order to light up a room. If you keep turning on the lights on all the time, even when you don't need it, you end up wasting electricity and you have to pay a hefty electricity bill. Same thing for our body. We can't be constantly generating red blood cells when we have enough. And actually, if we generate too much red blood cell, that can actually be a detrimental thing that I'll talk about in the next slide. Okay, so that particular message that tells our bone marrow cells to generate more red blood cells is the hormone called erythropoietin that I have mentioned earlier when I was talking about hematocrit. Okay, again, right here, erythro for red blood cell, and poietin, which means generation of. So that's why I say the names of these things pretty much tells you what it does, okay? And once you have these common terms down, medical terminologies will be a lot easier to handle, because when you get to nursing school, I'm sure you have to take a pathophysiology class or medical terminology class that require you to know all these different terms. So now is the best way to come up with something that can help you memorize all those different terms that you will continuously throughout your, your education towards your final goal, okay? So find some words that you can easily relate to and have a way to remember them instead of just remembering them by definition as is, okay? Okay, so EPO is a glycoprotein that is formed by kidneys and the liver when low concentration of oxygen is detected in tissue, and we call this condition hypoxia, okay? So hypo, okay, for too low, and cia for oxygen level. So hypoxia for low oxygen level. And there are 4 conditions that will specifically signal for the kidney or the liver to release EPO. There's got to be some kind of trigger. We cannot be releasing EPO all the time, which is bad for the body. So what triggers them to release EPO is any of these 4 things. All of them has to do with some form of hypoxia. The body is screaming for oxygen, and hence whatever it is, whatever the reason it is, there's just not enough oxygen for the rest of the body. So the only way your body can compensate and knows how to compensate is by making more red blood cells, so you can have more capacity to carry oxygen, okay? So these are the 4 conditions whenever you have anemia, which we already talked about the reason why they are in lack of oxygen. If blood flow specifically to the kidney declines, okay? So this is something we'll talk about when we get to the urinary system. It has to do with the arterioles and that's leading to the glomerulus around the kidneys, or even if the renal artery itself is blocked or something. Well, because the kidney is doing some of the sensing of oxygen level and blood flow levels, it goes by pressure, okay? We'll talk a lot about pressures this semester, but that's a main theme of the urinary system, is the different pressure levels that allow things to be filtered from our blood. So that's why it knows and can detect blood flow, okay? So when the blood flow to the kidney declines, it thinks that there is not enough blood, even if it was just a blockage at the renal artery that you're not delivering enough blood to the kidneys. It still thinks there's not enough blood because it only sees what it gets, okay? And that could trigger the stimulation and release of EPO, which can be if they check your hematocrit, yet your hematocrit looks normal, but you're releasing EPO, then that can be a pre-diagnosis clue as to maybe something is going on in your blood delivery to your kidneys, okay? So, a lot of times with diagnosis is a lot like solving a puzzle, okay? Is it this? Is it that? Can it be this? Can it be that? And you can eliminate your way down, all right? And the third way that the EPO is released is when oxygen content of air in the lungs decline due to maybe a disease, like, our famous disease right now, infection with COVID-19. What that virus does is that it creates lots of mucus within the blood, within the lungs, the respiratory airway, and also within the alveoli itself. And that's part of the reason why it's deadly, is because they like to infect in the lower respiratory tract. We'll talk about this when we get to the respiratory system, but there is a division of upper versus lower respiratory tract. Common codes, flu, things like that, things that are, less dangerous for the body usually infects the upper respiratory tract. Why those other scary diseases like SARS, COVID-19, and so deadly is because they like to infect also the lower respiratory tract, which then goes directly for our alveoli, which is critical for that oxygen delivery and exchange, okay? So sometimes because of that, then you have less oxygen available because those surfaces for oxygen exchange are gone, okay? And also in the cases of high altitude is also a reason why you can have less oxygen, because when you are at higher altitude levels, there's less oxygen available in those areas. So that's why then what ends up happening is your body will release EPO to generate more red blood cells, and hence you can still breathe normally when you're high up in Mount Everest or something, okay? And hence this is something we'll come back to when we talk about the respiratory system, why people that live in the high mountains can be superhuman, and they seem to be able to carry very heavy stuff, or for athletes that require a lot of respiratory and cardiovascular output, they do what's called altitude training. We'll talk about that when we get to the respiratory system. A lot of stuff is so interesting about A&P, and that's why I said this second semester is tough, but everything is so interesting. Okay, I guarantee you that. Okay. So the last reason why EPO is released is when respiratory surfaces of the lungs are damaged, okay? Like I was talking about with COVID-19 infection or pneumonia, those cases are when there's all these mucus build up within the alveoli itself, or sometimes with, we'll talk about emphysema, a disease where alveoli would fuse with each other, and instead of little holes, we have giant holes within the lungs, which, decreases that surface area for efficient oxygen exchange, okay? So this is just a glimpse into the respiratory system here. All right, so don't forget those 4 things that can happen to stimulate EPO release and why. All right, so there are 2 main downstream effects of EPO that leads to generation of more red blood cell. First, it will stimulate cell division rates in erythroblast stem cells, and also those stem cells, hematopoietic stem cells, myeloid stem cells we talked about. And it also speeds up maturation of red blood cells by accelerating the protein synthesis of hemoglobin. Okay, so just everything that leads to a mature red blood cell is sped up when you have EPO around. Okay. And this becomes an important factor if someone has had loss of a lot of blood from their body. They need to be able to regenerate enough blood. Of course, if the loss is too much that our body can't catch up, then they need a blood transfusion, and that's what emergency ambulances going to hospital is for. Sometimes we lose so much blood that our body system is not making enough red blood cell fast enough to keep us alive. But at maximum EPO stimulation, the body is going to try its best to do and keep homeostasis. So at maximum EPO stimulation, erythropoiesis can actually be increased by almost 10-fold in amount. Okay, and with understanding of how EPO works, like I said, usually from understanding how our normal system works, we can come up with ways to treat patients, or we can misuse those treatments. Okay, the original idea with EPO is if we can inject people with anemia with artificial synthetic EPO, then we can stimulate their body to generate more red blood cells, because they don't have enough to begin with. It was originally for patients with anemia, for patients with HIV, those patients that are just not, or even cancer patients, okay? Those patients that don't have enough red blood cell and they need more to supply oxygen to their body. Well, this also led to a misuse of the hormone called blood doping in healthy individuals, especially for the realm of Olympics or very competitive sports. Okay, blood doping is when a person ends up with more red blood cells than what it would have been in a normal individual. We talked about one of the major role of red blood cell is to deliver oxygen. And we learned from the muscle chapter last semester that in order for us to have actively contracting muscles, we need a lot of oxygen to generate a lot of ATP. And hence, if one can have more red blood cells than they usually do, technically speaking, one can harbor more oxygen and as a result, able to last a bit longer or result in stronger muscle contraction for you to win that race as first place or win the gold medal in Olympics. Okay, there are 2 ways blood doping can be done. One way is by blood transfusion. Okay, so make sure you remember blood doping, and one of those ways is transfusion, where before a race, athletes can remove blood from their body or someone else's body if it matches in blood type, freeze it down as a reserve, and then when the race time comes, they will transfuse those extra blood reserve back into the body, which can almost result in a 50% increase in red blood cell concentration. Now, the other way of blood doping is by the way of what we just learned about, EPO. And there are, like I said, there are synthetic version of EPO that has been manufactured for the purposes of treating patients, but it has also been sold in the market for incorrect purposes, such as gaining more ability in a race. Okay, so they will inject EPO into their body and artificially induce the red bone marrow to make more red blood cells. And of course, a lot of times, doing something that is not normal or artificial for the body oftentimes come with consequences. There's almost never, cheating in a sense. The consequences will come back to you. Okay, cheating in any form. Okay, so that's why, cheating is never the right way to go. It ends up catching up on you. In this case, you can lead to death. Okay, so it can actually be a very deadly consequence. What happens is, remember, our blood is related to the entire cardiovascular system, which one of the main organs in charge of pumping our blood around the body is your heart. And our bodies are designed to only handle a certain volume and viscosity of our blood. And that's why our hematocrit levels are usually maintained within that 45% range. Or else, why would there be a normal quote-unquote, hematocrit percentage? All of us can have 60-80% hematocrit. Why can't we have that? It's because our cardiovascular system is designed to only handle that range. If we put it above that range, we will put a lot of stress on the heart and blood vessel as well. The fact that blood now contains so much more blood, so much more cells, red blood cells specifically, can put a lot of extra strain on the heart to pump blood into arteries and hence increase the likelihood of heart attacks, stroke, and also formation of abnormal blood clots, which can ultimately lead to stroke and heart attack. Okay, so imagine all these extra red blood cells on top of that in the athlete's body. Their heart is already pumping very hard to allow the athlete to race. Usually they're already pumping above 100 beats per minute, and yet there's all these extra viscosity coming from all these extra blood cells. And in addition, on top of that, the athlete's body is in dehydration mode because of all the sweating that they're doing at extreme levels of muscle activity. All of these things turns, one article puts it that way, it turns blood into mud. Okay, so you can think about trying your heart trying to pump mud through your cardiovascular system. And that's why there were incidences where young cyclists, a few of them in their 20s, have died in a race because of that. Okay, so thankfully this practice has been banned and is illegal now, but not only because it's not fair for the competition itself, but it really puts the athletes' lives at risk. You're a perfectly young, healthy individual, and probably you are very, your body systems are working very fine, or else you can't be a good athlete. But you're putting all that at risk when you go through this cheating method. Okay, is it really worth your life for that one gold medal that one may win at the Olympics? One can't even enjoy the fame anymore if you end up dying. So it's really not a good choice. Anyway, so as a last note on the process of erythropoiesis, the body also needs adequate amounts of amino acids, iron, vitamins like B12, B6, and folic acid in order for erythropoiesis to proceed normally. And you can imagine why amino acids and iron is important, because that's what forms the bulk of hemoglobin as a protein structure itself. Okay, so it has been an hour, so we'll take a break here for part 1 and we'll come back in part 2 of the lecture.